taken away all of these light capturing or photosynthesis processes. So what we do essentially is study these at a fundamental level. So we do quite a lot of work on photoreceptor function, kinase signaling, substrate identification, as I'll talk about today. And essentially what we're trying to do is determine how these receptors control differential cell expansion and organelle movement. And I'll tell you about one of those components in today's talk. And we're doing this by looking how this love motif, this light sensor, controls these processes. And we've also hijacked this to make sort of synthetic photoswitches with other lab groups. So we've got projects whereby we're controlling plant physiology through optogenetic manipulation, which I don't have time to talk about today, but also we've engineered these to be alternative fluorescent reporters to GFP. But I want to focus back on the movie here, which is the growth response of Arabidopsis seedlings to directional blue light. And I want to talk mostly about this protein here, which Tatsuya, who's after me, will talk about in more detail too. So this is non-phototropic hypocaudal 3. I've kind of taken a reductionist view here in terms of the phototropic signaling. So I'm just going to focus on this component here. So as a protein, it was one of the first proteins that was identified from genetic screens for phototropism mutants in Arabidopsis. It is a BTB-containing protein, so it's been linked to E3 ligase ubiquitination. But currently the targets for this are poorly understood. FOAT1 has been shown to be a target for MPH3 ligase activity, but the function of this is not fully understood. There's also a coiled coil region here that's involved in associated with protein interactions and signaling, the C-terminus. So if you look at the mutant here, the MPH3 mutant, you can see it's devoid of phototropic curvature. It's also devoid of root phototropism, but its gravitropic responses are completely normal. And Christian's really nicely summarized the figure here showing that a light gradient of phosphorylation is needed to stimulate this differential growth. So as indicated here with receptor autophosphorylation across the hypocaudal, and this in turn gives you a differential accumulation of auxin on the shaded side, which leads to cell expansion and differential growth. But this occurs by, as yet, an unknown mechanism. We still don't know how this is regulated. So if we look at MPH3, it has a couple of characteristics at the cell biological level. It's plasma membrane associated. It associates with phototropin-1 at the plasma membrane. And if you look at the relocalization properties, you can see here this is seedlings expressing MPH3 tagged with GFP. So you've got membrane localization. The laser scan to excite GFP is also exciting phototropin. So you get a concomitant relocalization of MPH3 after it's been activated by phototropin, and it relocalizes into these intracellular aggregates or condensates, as they've been called. And if you track these condensate formation across the hypocaudal, you can see that you get a differential gradient of this condensate formation being formed. So on the irradiated side here, the site close to perception of blue light, you've got more condensate formation versus the shaded side here. So this is consistent with a receptor gradient being formed across the hypocaudal and being sustained throughout phototropic signaling. So when we typically do phototropism, we're continually stimulating these seedlings with blue light for a duration of 1 to 2 hours. So you need to establish this gradient and sustain it for that time period. So we know that MPH3 is a substrate for FOAT1 kinase activity. To do this, we use a system whereby we engineer phototropin-1 to gatekeeper the photoreceptor so it incorporates an ATP analog. So you're introducing a thiophosphate instead of a regular phosphate into the protein itself. You can make the protein quite easily in vitro. And here's a Western blot that shows essentially light-dependent thiophosphorylation of phototropin as its hallmark activity for this receptor protein in the system. And if you co-express with MPH3 on the bottom here, you can see that you've got light-dependent phosphorylation of MPH3 in the presence of phototropin-1. And if you mutate a serine residue that's on the extreme C-terminus of MPH3 that's part of this motif here, RXSIS. So it's this penultimate serine here, serine 744, that's the site for photoreceptor phosphorylation. So when you mutate this to alanine, you abolish this phosphorylation. So we know through our functional characterization of this site, it's required for MPH3 localization. So here's transgenic seedlings. You're expressing wild-type MPH3, so it relocalizes in response to blue light into these condensates. Here's the mutant here, so you get no relocalization of MPH3 when you mutate the FOAT1 phosphorylation site. And this is an artificial form of MPH3 where we've taken that C-terminal sequence, the RXSIS motif, replaced it with what's called an R18 peptide. So this is a peptide that can bind 1433 proteins, and I'll come to what that means there. And what we get here is that if we replace the site, we get constitutive relocalization of MPH3 into these condensates. And the reason for that is that that FOAT phosphorylation site is a binding site for 1433 proteins. So here we've got data by far western blotting to show that if you IP MPH3 from extracts and probe with GST 1433, you can see you get strong binding of 1433 proteins to MPH3 in the presence of light treatment. And when you have that serine 744A mutated, you lose this ability to bind 1433 proteins. And conversely, in this R18 peptide version, you have a constitutive ability to bind 1433 proteins, which matches with its ability to relocalize constitutively from the plasma membrane into these condensates. So if we look at the functionality of this, here we're looking at the phototropic response of transgenic seedlings compared to seedlings expressing the wild-type MPH3 form in the MPH3 mutant background. So you can see that the serine 744A mutant and the R18 mutant here has severely compromised phototropic curvature responses compared to the wild-type. They are still responsive, and that's something that I want you to keep your mind on as I go through the functionality of this moving forward. So essentially, as a recap here, you need a sort of light gradient for full establishment of phototropism. If you've got an MPH3 form that is either can't form a light gradient in terms of its ability to form condensates, you have severely reduced phototropism. It's still functional to a degree. And likewise, if you've got constitutive 1433 binding, you've got a small ability to have this phototropic response. So what does this mean? So how do we think MPH3 is working? So in the dark, we know that MPH3 is plasma membrane localized. We know from Tatsuya's work that MPH3 is also phosphorylated at other residues within the protein, within the N-terminus. And phototropin light sensing essentially is the initial trigger that will essentially activate MPH3. So we've got some sort of light-dependent mechanism between MPH3 and FOAT1 that causes this ability to form an auxin gradient. And what I'm trying to do here is just rationalize our phenotypes of our serine 744A mutant that still has an ability to form a phototropic response, albeit poorly. But we do know that FOAT1 phosphorylates at this residue here, 744. It binds 14-3 proteins. And this also leads to dephosphorylation of these sites in the N-terminus. As you can see here, that's easily tracked by electrophoretic mobility shifts in the MPH3 protein here. So you increase the electrophoretic mobility of MPH3 when you remove these sites, and you see this nice band shift, and this leads to condensate formation intracellularly. So what we think here is that this might be a way to rapidly switch off the signaling system because it's highly dynamic. So through changes, rapid changes in phosphorylation status, essentially what we can have here is to restore this back to the actual native form. Is that my bell already? That's 10 minutes. You scared me. Or is that just to wake me up for my jet lag? Okay, so you can actually, on the reversal of this, you can see what we've got here in darkness is a reversal of this. So you need to rephosphorylate these sites in the N-terminus, and that's because you need to dephosphorylate the phosphorylation site to reestablish membrane localization. And why do we think this is happening? We think this is a way to amplify the signaling system, to amplify the gradient signal. that's just a hypothesis that we have at the moment. So there's two main questions here. We still don't know how auxin gradients are being established through MPH3, and also we don't have an idea of what the sort of regulators here for the phosphorylation, rephosphorylation of, and dephosphorylation of these components here. So we wanted to check this out, and a candidate phosphatase that we were interested in was the PP2C family. In Arabidopsis, there's 80 of these, so it's a challenging family to dissect. And essentially we were interested in these because some of these clade members have roles in cell expansion. So if I focus you down here to clade D, E, they have been reported to play a role in cell expansion responses. Some of these clades as are yet uncharacterized. So what we thought we would do is see if any of these played a role in the phosphorylation status of MPH3. So we're going to focus on clade D, E, and L here. So we've got mutants whereby we're going to check whether they're altered in phosphorylation status. So for this experiment here, what we're doing is that we're treating seedlings with light, and then we're putting them back in the dark and assessing S744 phosphorylation status over a duration. So you can see here the phosphorylation status of S744 is basically dephosphorylated and restored after 15 minutes. And there's a triple mutant here of clade D. There's a sort of little small effect that you're seeing, a sustained level of S744 phosphorylation. But most notably here is one of these clade L members, that's PPC2C19. You get quite a substantial sustainment of S74 phosphorylation in darkness compared to the wild type. And this is also reflected by a rephosphorylation of these other sites in the N-terminus here. So if we're looking at the electrophoretic mobility shift, we can see the triple D clade mutant here and the PP2C19 mutant here have delayed sort of rephosphorylation compared to the wild type. So it seems that you need to dephosphorylate S744 to get rephosphorylation of these other sites. And you can monitor this in a little bit more detail. So we focused on these clade L members. There's two, they're uncharacterized in terms of function in Arabidopsis. And most notably here, over the kinetics, you get a really marked sustainment of S774 phosphorylation when you make the double mutant between clade member, these two clade members. So there's PP2C19 and PP2C35. This is specific for MPH3, so we can also do the same sort of experiment and look at FOAT1 dephosphorylation in darkness. So I've told you that FOAT1 is autophosphorylated in response to blue light. We've got a phospho-specific antibody that can monitor one of these sites. If you put them in darkness and wild type, that site will be dephosphorylated after about 30 minutes. And these mutants show no altered kinetics in terms of the dephosphorylation for that site. So what we're looking at here appears to be specific, at least for MPH3. What about the phototropic response? If we have a look at the phototropic response for these mutants, it tallies up with what we see in terms of changes in MPH3 dephosphorylation status for serine 744. So most notably, PP2C19 and the double mutant here show marked defects in phototropic curvature. And we see this over a range of light intensities. So if we go to lower light intensities for phototropism, we still see these defects. If we increase the light intensities, we still see these defects. So it seems that by modulating the phosphorylation status of serine 744, we can alter phototropic responsiveness. As we were looking at the hypocaudal phenotypes for these mutants, we also discovered that the double mutant, and in particular here, it displays a gravitropic phenotype. So that's visually shown here in the seedlings and quantified here. Quite interestingly, we don't see any alteration of root gravitropism as well. So when we make a double mutant between these two PPC clade members, we do see hypocaudal gravity defects as well. So just to summarize there, I showed you this model already. This seems to place these clade members here, whereby they're required for regulating the phosphorylation status of serine 744, so they can dephosphorylate this residue and then stimulate rephosphorylation at the N-terminus through an unknown mechanism or protein kinase that we still don't know what the molecular identity is. And this is consistent with what we see if we co-express one of these PP2Cs with MPH3 in tobacco. So MPH3, this is RFP MPH3. You can see it relocalizes as we would expect in response to light treatment on the left. But if you co-express with the PP2C35 here, you block this relocalization, and that's simply because you're dephosphorylating the serine 744 site. So you're actually you're mimicking that mutant serine 744A and blocking the light-dependent photo movement from the plasma membrane. So just in the final stages of the talk, is this only applicable to MPH3, or does it apply to other members of what Christian referred to, this NRL family? So this is the MPH3 RPT2-like family in Arabidopsis. There's 33 members. 4 of these members we know function downstream of photes. I've told you about MPH3. There's the closest homologue of MPH3, DOT3 in Arabidopsis. We know from our work, I can't show you this today, but we know it overlaps in function with MPH3 to regulate leaf positioning and expansion. NCH1 is one of these family members that regulates chloroplast accumulation movement, and RPT2 seems to be involved in all of these processes. But I will highlight that the biochemical function of these proteins, we still don't know what they're doing. So I'll just end with some data on NCH1. So we know that NCH1 is also a substrate for FOAT1 phosphorylation. So analogous to what I showed you before for MPH3, you can co-express FOAT1 with NCH1, and you can see nice trans-phosphorylation between FOAT1 and NCH1. And you can abolish this if you mutate that C-terminal residue, which has that serine phosphorylation site. So in NCH1, that's serine 602. And if you have a look at these Clade L members, if we take a look at the double mutant here, if we can have a look if there's an impact on phosphorylation status. So here we're using light-grown seedlings because NCH1 is involved in chloroplast movements. We can also quickly look whether there's an impact on phosphorylation status. So here we're just giving a blue light treatment. You can see with a phospho-specific antibody here, you can measure light-dependent phosphorylation of NCH1. It appears to be also phosphorylated in the dark here. But what you can see here in the PP2C double mutant, you've got an increased elevation of serine 602 phosphorylation. And if we look at the ability to kind of dephosphorylate in darkness, if we look at NCH1 here using that antibody, serine 602, you also see there's a mobility shift here, suggesting there's an analogous system here with MPH3. There's other sites that get dephosphorylated in response to light treatment. It's very rapid in the wild type here, so it's restored after 5 minutes. But if you have a look at the double mutant here, you can see that you've got highly sustained S602 phosphorylation when you take these members away. So does this have any impact on chloroplast movements? Well, together with Ustina Wabus at Krakow, we have done experiments whereby we're looking at red light leaf transmission here. So you can use this to track chloroplast movements. So if you have low light intensities of blue light, chloroplasts accumulate to the upper cell surface and reduce the ability to transmit red light through the leaf. So you can detect this through a photodiode. And conversely, under high light conditions, you make the leaf paler because of the chloroplast avoidance movement, and hence you increase the ability of red light to be transmitted through the leaf. So you can use this as a tool to track chloroplast movements. And what we're looking at here is chloroplast accumulation movement. Looking at red light leaf transmittance. This is the last slide, whereby if you look at the wild type here, blue light induces a reduced red light leaf transmittance, and in connection with the results that we also found for phototropism, the PP2C19 mutant and the double mutant here have reduced chloroplast accumulation movement in response to low intensity blue light. But we don't see any response defects under higher light conditions. So under higher light conditions, you have the chloroplast movement avoidance movement response. So blue light increases red light leaf transmittance, and this is completely unaltered in the mutants. So it seems to be something that's specific to chloroplast accumulation response. And I just wanted to note here that it's FOLT2 that controls this response. And to date, there's no NRL protein that's been associated with the chloroplast avoidance movement response. So just to summarize, we've got this clade L family of PP2Cs that appears to be regulators of early seedling establishment. And this is via the effects of hypocaudal gravitropism, hypocaudal phototropism, chloroplast accumulation. And they seem to be doing this by altering the phosphorylation of specific NRL proteins that are involved in phototropin signaling. We've also looked at other family members. So RPT2 is another member that I've mentioned. Essentially, we see impacts of phosphorylation in the PP2C 35 double mutant. We also see sort of large impacts on abundance as well. So these PP2Cs are also affecting other NRL members. So just to thank the people in the lab, much of the work that I talked about today was by Stuart Sullivan. Dimitra here, she's working on DOT3. I didn't have a chance to show you some of the data here, but the PP2C project was initiated through our collaborations here with Noriyuki Suetsugu, Takeshi Higa, and also Iji Goto in Japan. And I'd also just like to end with this slide here. Some of you might recognize these people. I've been very lucky when I did my postdoc in Winslow's lab to meet these guys and visit Japan several times. So Masamitsu Wada, Satoru Tokutomi, and Kenichiro Shimazaki. And here's them enjoying a nice summer's day at Loch Ness in Scotland. So the weather's always like this in Scotland, I promise you. So I'll finish there and happy to take any questions. So thank you very much.